Welcome everyone to this month's Discovery Series lecture. Really excited about the talk. There's a lot of reasons to be excited about this space. So the goal of the Discovery Series is really to share cutting edge research that aims to improve health for people here in Missouri and beyond. And the speaker is going to share ways to do that and really amplify connections and collaborations. Before we introduce our speaker, I have three quick announcements to make. First of all, the way the schedule has worked out, our next Discovery Series is actually next week. So please join us to listen to Dr. Mark Towler talk about his work taking a new bioactive glass that prevents excessive bleeding. So next month, we'll actually be in Rolla for the in-person talk. We'll have live streaming here, and we'll also be sharing this online by Zoom. Next reminder I want to give everybody is mid-October is the deadline for the NextGen postdoc program. And so we're really excited to kick that off. So please get the word out and share about that program. Lastly, one of the most important parts about the Discovery Series is having the chance to dig deeper, ask questions. And so to make that opportunity equally available online, people here in the room, we use a process called Mentimeter. We use the Mentimeter to ask questions. So please enter your questions using Mentimeter. If there's a question on there that you really like, you can upvote it to the top of the list. If you're online on Zoom, you can also use the chat, but we really would prefer Mentimeter if at all possible. So before I hand off the mic, I want to introduce the person that's going to introduce our speaker, a friend and a colleague, collaborator, John Ruth. He's a Jefferson City native. He's alumnus of Mizzou's College of Business and also the football program. He's a co-founder and CEO of Build Asset Management, and he leads the Genetic Autism Alliance, which is funded by the state of Missouri and aims to improve treatment and diagnosis for individuals impacted by autism. And so with that, I'm going to hand off and let him do the introduction of our very exciting speaker.
M-I-Z. There you go. That's the right reaction. I get to give my good buddy trouble whenever he gets here. You'll notice him because he's about 6'6". So you'll help me out in giving him some problems when he gets here. But I'm going to talk about Curtis Gregory for a moment. So I'm going to save, because he's late, he's ruined my intro. But Curtis and I were teammates and roommates here at Mizzou. And I'll come back to that here in just a second. Dave talked about the Genetic Autism Alliance. And so before I introduce Dr. Weiss, I want you to think about three numbers. And I want you to envision these numbers and think about these as Yael is talking today, and take these with you, hopefully, out of the room. The first is the number 36. Second is the number 10. And the third is 0. And those are not the pick three numbers for this evening. So what are those? Well, before I tell you about those, let me tell you why I'm here. I'm Camden's dad when I'm in this building. Camden is an 11-year-old who lives with a rare genetic condition. It's numerically referred to as SYT1 disorder. Its nickname is Baker-Gordon syndrome. And, Camden is like any other middle child. She's a nuisance to her younger sister and sometimes a nuisance to her older sister. And she's brought a great and unique joy and also a challenge into our life. And, just as Dave mentioned, school, which was the main reason I was here. The NFL was not calling me, but the things that you learn and hope that you can apply in your life from sports, those hard moments that we talk about, or the grit and the grind of, football, combined, I think, with faith for us said, what are we going to do about this? And Yael and I are friends in industry, but also online. I shared earlier this year some quotes that spoke to me in this field. Yael mentioned she liked one of the three in particular, and since it's her second time to Missouri, I thought it'd be appropriate to mention a famous Missourian, Mark Twain, who is usually credited with this quote. And the quote is that the two most important days in your life, the day that you were born and the day you figure out why. And this comes back to my buddy Curtis. So we're all going to boo at Curtis or something when he comes in here. So what I wanted to say is what I thought when Curtis and I were living together, almost 2.5 years, became really good friends, that if we came back to campus at some point in the future, we might talk about the good old times of football. We were on the 2007-2008 team, which was the highest ranked team in school history to date. Hopefully they'll beat that this year. And so we thought, well, maybe people will talk about the good old days of football, and we'd have some fond memories about that. And what I would submit to you, my new loftier goal is, I hope people will talk to us about coming here, treating and ultimately curing a very difficult, rare disease. And that'd be an even better legacy than what we can leave on the field. So that's very much my hope. Curtis has been an important part of the story. He currently serves as a state representative and running for state senate. He's been a huge supporter of the GAA and the University of Missouri. We also have some other distinguished guests with us here today. Representative Rudy Veit, who's my representative, serves in the state capitol. My senator, Senator Mike Bernstetter. A dear friend of the university and of mine, Tom Rackers, and John Ratliff, who serves with Palm Strategies and helps some of these folks get elected and also work on unique policy challenges in the state. So thank you guys for coming here today and spending time with us. Back to the numbers. How many folks, by show of hands in the room, know someone, have someone in your house, have a colleague or a friend who is impacted by a rare disease? All right. That's a lot of hands.
Mm-hmm.
So when I went to the Capitol to talk to our elected officials about this problem and the unique opportunity to work on that here at MU, I was nervous. I don't get nervous usually speaking to people, but I was nervous about this. And my nerves calmed down instantly, because the first office I walked into, yes, Camden has SYT1, but she also has an autism diagnosis. So that might be easier to explain in an introductory meeting. And so I have a daughter who has autism. I'd like to talk to you about it. And people would say, oh my gosh, so does my child. Come on in. Or so does my staff member. I have a constituent. These numbers, the reason that these numbers are so important and impressive, 36 is the rate of autism diagnosis in the United States. It's 1 in 36. 10, even though we talk about rare diseases being these obscure, less than 200,000 occurring challenges in the medical field, in the aggregate occur at a rate of 1 in 10. It's really a wonder that any of us are put together the right way at all, because these are often just single monogenic problems that present. In 0, what I was really floored by when we went and had our autism evaluation and services, I asked those doctors, how often are children receiving an autism diagnosis and then immediately sent down the hall to get whole genome sequencing? And fortunately, because some other folks I want to talk about here in a second, that's happening some here at Mizzou. But as a national standard and a best practice, the answer is 0. It is not a standard of care in the United States. And I thought my head was going to explode. Because when we got our SYT1 diagnosis for Camden here, we were told she was 1 of 9 in the world. And I said, I've never even won a raffle at a luncheon like this, let alone been 1 of 9 of something. That's impossible. I'll bet you if we just tested more folks, we'd find more of her. And how do we know what to treat or what to work on if we don't even know what the problem is? And so, again, very fortunate. Remember everyone on 3, boo, 1, 2, 3, boo. Curtis, I pumped you up, man. This is Representative Curtis Gregory, and also my very good friend. He's a part of a team that supports everybody in this room and the reason we're here today.
I got lots of college stories on that guy.
As do I, but I'm not running for office, so you might hold yours. So listen, we're here today because I think, Curtis, here's what I was telling folks. A great story and a great reason to be back here at MU. Certainly we're proud of our school sports and programs, but wouldn't it be great if we could say we accept really tough challenges here at the University of Missouri and we can do something about them. And I think that's a great mantra for the state that feels, so much like kind of the American spirit. And so I'm very proud of the black and gold, but especially because of what's taking place in this building and around campus. So hats off to all of you who are working on these problems. One more introduction before I introduce our guest speaker. We wouldn't be here at all today if not for two people. And the first is Dr. Paul Carney. Paul saw us as a pediatric neurologist because we were interested in looking at an off-label drug that we thought might have some impact. Paul looked at us and our problem. He sized us up a little bit and he said, yeah, we could probably look at this drug, but why don't you want a cure? I said, I didn't know that was an option. Which door do I go through for that? And Paul planted this seed and said, you know, we live in a pretty wonderful time. This is a challenging disease, but we can actually do something about these challenges in the point in time that we live in. And that wasn't always the case. And you ought to go find people who are working on these and see if we can come back and incubate some bigger ideas here. So Paul, thank you for inspiring that confidence and being such a big part of our team today. One of the first people Paul took note of was going a little bit against the grain. I think you had read some notes from Yael in a conference. He said, you guys are looking at this the wrong way. We got to do this. And Paul reached out to Yael, and pretty soon Paul said, you got to go see Yael. So I did. And boy, Yael told me exactly how it was first time we met. And so she's going to share some good ideas with you here today. But Yael and I have become good friends. She is known as the CEO of Mahzi Therapeutics, which focuses on ultra-rare disease therapeutics, especially neurodevelopmental conditions. But more importantly, she's a champion of rare disease. And there's just not enough of those in the world. We need all of them. You'll see here in a second, she's not as big as Curtis, but she plays in the heavyweight arena, just like you saw my friend Curtis walk in, who's a part of the team here too. So I'm going to stop talking now and turn this over to our important speaker, but please help me in welcoming Dr. Yael Weiss.
I have a microphone. Is my microphone working? Is it working? Can you hear me? Yes. Okay, great. So I won't use this one. Well, first of all, I want to thank everyone here. I'm very excited to be here. It started with Paul, who introduced me to John, who introduced me to Dave, which actually culminated in me being here today. And a lot of great meetings. I'm learning a lot about how great this university is and how much we can do together to develop therapies. I will go through a very varied talk, try to cover a lot of topics. I know there's a mixed audience here. So if you have any questions while I'm talking, if you feel that I'm shifting to a foreign language to you, please raise your hand and ask. Make sure that I can clarify whatever is not well understood. I do want to say a few words about how I got to be the CEO of Mahzi Therapeutics for the benefit of those that still haven't chosen a career. I actually started with a PhD in molecular genetics many years ago, and my PhD was on Down syndrome. And I realized, gosh, these genetic conditions, how can it be that just one change in one letter in your DNA, or a little slice that is taken off, or a little slice that's added in, results in such a different clinical presentation and changes the life of a patient and their family. And I really wanted to investigate how this happens. So I decided to go to medical school, and I earned an MD. And when I was in this MD PhD place, I thought, maybe the most interesting place is to be where we actually translate work from the bench into the clinic. And why don't I just start developing drugs? And this is how I found myself in the pharmaceutical industry, biotech industry. So whoever is passionate for translating basic research, this is the place to be, and highly recommended. And Mahzi specifically was started because of parents like John, and I'll tell you a little bit more about Mahzi. So even though the title is Developing Gene-Based Therapies, it's really the second part of the title that I want to focus more on, the joint effort between biotech, academics, and parents and patients. This is the critical triangle to succeed in drug development, especially in rare diseases. So this is Mahzi, the company, a young company that was founded 3 years ago. You see Mahzi in the center. We're all rare disease experts, all have experience in developing therapies for rare diseases. But we cannot do it without, on the top, the advocacy groups. On the left-hand side, the academics. So just like John, many parents, many patients turn to academics to develop a therapy for themselves. But at a certain stage, they exhaust the options. They don't know how to develop a therapy, and they don't have the resources to develop a therapy. So they get stuck. So this is where Mahzi comes in. We take over the programs that were invested in an academic research lab and move them into clinical trials and hopefully to a commercial environment where the drugs will be accessible to patients. And we do it with diagnostics companies that I'll talk about in a minute, and of course with different vendors that help us make the drugs and develop them. So if you plan to fall asleep after the lunch, these are the key messages for my talk. First of all, the most important thing is that the genetic diagnosis landscape is changing, and the more gene-specific disorders there are, the more gene-specific therapies there will be. So it's really critical to get a genetic diagnosis, and I know John is very passionate about it, and we'll talk about it a little bit more. Drug development is not for the faint of heart. It's difficult, there's a lot of failure, and you have to really persist in order to get a drug from an academic bench to the market. It fails early in animal studies, it fails in clinical studies, but we have to go back and learn from our failures and try again. Proof of concept, so what is done here in the university, a proof of concept in cell lines or in animals, that's only the beginning. The path is very long. I'll talk a little bit about what happens after it leaves the labs. And then the partnerships with the academics, with the patient groups, and with the industry are critical. There are multiple tracks of work in parallel, so you can't just start a work in the lab and expect that the drug will be in the patients in a few years. There are a lot of other things that you need to do in parallel. Research should be very translational. Unfortunately, most universities, most academics need to publish, need to write grants. Making drugs is not always the first thing on their list. So how do we shift the thinking and make drug development and drug discovery more motivating for academics? That's a great question to the university. I personally cannot answer it. The goals need to be aligned, and there needs to be a sense of urgency. John Camden is 11 now. She is waiting for a therapy. She can't wait much longer. We don't know how old you can be in order to benefit from a drug that is injected into your brain, but we do know that the earlier, the better. So kids, the young kids will probably benefit more than older kids, and we want to get into patients as soon as possible. So we can't wait for another paper, another grant. We need to do it now. So I'll cover a little bit on genetic diagnostics, a little bit about what a profile for a drug should look like. When you're thinking about, I'm going to develop a drug, or I want to be involved in drug development, or I want to help a drug be approved, this is what I should be thinking about. What type of modality am I using? Is it going to be a pill that I'll take in my mouth? Is it going to be something that's injected into my spine? Is it going to be something that's injected straight into my brain if it's a brain disorder, et cetera, et cetera? What needs to be done after the animal studies, and what does teamwork look like? So I'll start with this, and this is really related to the diagnostics part. We are really in the midst of a precision neurology renaissance. If you had a child that was born maybe 15 years ago, 20 years ago, they would likely get a diagnosis of, oh, this child has epilepsy, or this child has autism. And that was it. There wasn't much beyond it. Maybe there was a 3-name disorder that is based on the people that first described it, like, what's yours? Baker Gordon, Dr. Baker and Dr. Gordon first described it, but nothing beyond that. Now, if a child is diagnosed with a SIT1 mutation, they will call John, and John will say, guys, we're working on a genetic therapy, and hopefully there will be one soon for your children. So a very, very big difference in approach versus what was true 10, 15 years ago. If you look on the right-hand side on the graph, this is actually describes the genes that are related to epilepsy. It's just the one that I found that I think is very telling. If you were born, if you look at the graph before 2011, 2013, you likely, even if you were sent to a geneticist and were sent to get a genetic test, you likely didn't get a diagnosis because the genes were not known. If you look at what happened from 2013 to today, hundreds of new genes were identified to be associated with epilepsy. And now, if you have a child that seizes and you get a genetic test, you likely know what the gene that causes this epilepsy is, and you can look for a genetic solution. So it simplifies life, it shortens diagnostic odysseys that John went through and mentioned, and helps us as drug developers understand disorders better and develop therapies for these disorders. So there are actually thousands of patients out there, probably older kids, that were put a tag of seizures, autism, developmental disabilities. No genetic test was ever done. There are probably thousands of patients that have a genetic diagnosis and we just don't know about them. Now, why am I so interested in finding these thousands beyond understanding the disorder? Pharmaceutical companies, they like to make a lot of money. They make a lot of money from obesity, right? That's the billion-dollar thing that everybody's now looking into. But when there are only 9 patients in the world, 90 patients in the world, 900 patients in the world, no company is interested. No one's going to make money out of it. So we need to find more patients to make it more commercially attractive as well. This is why it's so important. I'll give you an example, and this is for Dave, whose expertise is SMA. I wanted to give an example of how we choose which treatments to develop for different disorders. So I took SMA as an example because it's a relatively well-developed disorder. People understand it, and there are different types of therapies available for it. So it's for those of you who don't know what SMA is, and I assume half the room does because they've already educated you, it's a rare neuromuscular disorder, and it results in loss of motor neurons and results in very progressive muscle weakness and death. It's caused by mutations in a gene called SMN1, and until 10 years ago, there weren't any therapies for SMA. In the last 10 years, there have been significant advances in treating SMA, increasing the lifespan of patients with SMA, and improving their quality of life, and as a result, the quality of life of their families as well. So I'll describe the 3 different therapeutic modalities that are available for SMA as an example of what we would do if John came to us and said, okay, I have a child with a mutation, what type of therapies are available to me? So for SMA, there are 3 types of therapies as examples. On the left-hand side, yes, there's a small molecule. What's a small molecule? It's just like aspirin. It's a pill that you can take in your mouth or something you can drink. You usually take it every day. It's just a small chemical. You can see the chemical structure of the specific SMA therapy. It's oral, and it's very easy to take. It has some limitations, but it's a great solution for patients who can actually swallow oral drugs. In the middle, you will see a different therapeutic modality called antisense oligonucleotides. These are small pieces of DNA or RNA that are given into the blood system. They penetrate into the brain, and they either activate or inhibit genes from making the protein. And then on the right-hand side, you'll see the newest modality, which actually the order is a little reversed for SMA. That was the first therapy that was approved. It's a gene therapy. What is a gene therapy? A gene therapy is when you take a gene that's missing or mutated, you pack it into a virus, you give the virus infects the cells that it recognizes. In our case, when we work on neurodevelopmental disorders, the virus needs to recognize the neurons. It goes into the neuron, it sits there, and it makes the normal protein, restores the function of the normal protein. So this is the most complicated therapy. It's given only once because the assumption is that the virus goes into the cells and stays there, so it doesn't go away, and it makes the protein almost forever. So the idea is that you're moving from an oral daily pill to a therapy, the antisense is given every few months, to a therapy that hopefully, we don't know yet, will be a one-and-done therapy. So this is a little bit more for the scientists in the room. How do we choose which modality we want to use for different therapy, for different disorders. So the small molecule, it doesn't treat all the mutations. It's a hit or miss. If you can find a small molecule, that's usually great, but it's very rare that a small molecule will fix a genetic mutation. In gene therapy, it treats only disorders where the protein is missing and you need to replace it. So you have only 50% of your protein and we want to give back 50% to restore the function. And antisense oligonucleotides can work in both situations where there is missing protein or there's protein that's there but it's not doing its function correctly and you need to get rid of it. So these are the three different options when we think, okay, am I missing something? Do I have too much of something? Let's think which is the best modality for each situation. As I said before, small molecules are given orally in most cases or by injection. They're the easiest to take as a patient. You know, all of us, well, I guess I'm showing my age here, but I have a little pill box next to my bed and I take pills every day, so that's the easiest to go. The gene therapy, as I said, one and done. Injection usually for CNS disorders straight into the brain. So it's a procedure that you have to go through, but it's a one and done. And then the antisense oligonucleotides are given 4 times a year because they break down and you need to replenish them, and they're given into the spine. The small molecules, if they are designed correctly, they will go into the brain. That's not a problem. Gene therapy, the viruses, as much as we know viruses are very sneaky, we still don't have a way to make sure that the whole brain gets the virus. So we cannot treat conditions that need 100% of the cells affected. We can only treat in cases where it's enough to treat only part of the cells, which is a limitation, but in some cases that's the only solution, and many families and patients opt in for it, even though it is limited. And the antisense are well distributed. I won't go through the whole table. I'm happy to share this afterwards. But just at the bottom, from a biotech perspective, I started raising venture money. And my 2 first programs are AAV gene therapies, which are extremely expensive. Even before we treated a patient, we already spent $10 million on making virus. So that's really, if that's an option that we would choose, if that's the only option, because to spend $10 million and then to get into a clinical trial and realize that it's not working is really sad for me and for my investors. But we are developing 2 gene therapies. The ASOs are not as expensive as gene therapies, and the cheapest are the small molecules. Again, another semi-scientific slide, and ask me questions if I'm talking way over your head, but there are different things we need to know before we choose a therapeutic modality. And it's not obvious that one is more suitable than the other. We really have to think, okay, first of all, not everyone can swallow pills. So maybe pills are even out of the option here. So we need to look at the ones that are injected or given to the spine. That's like a way broad consideration before we go into which modality we're going to use. But we need to know a set of things that really will help us design a therapy and prove that it's working before we go into clinical trials. And how do we do it? We do it by analyzing the gene and what does it do normally? What does the protein do normally in the body? And where do we need to replace it? That's really important, right? If we need to replace it in the liver, then yeah, we won't deliver it to the brain. But in some cases, there's a protein that works in the brain, but also in the liver and also in the muscle, which means that we need to get to the whole body. And that will be a completely different approach than just injecting to the brain or just to the liver. So we go through this list of different characteristics, and we try to understand what's known and what's not known. And then we prioritize what we need to complement in order to get proof of concept that the drug is working. Again, I don't want to go through the table. There are a lot of very scientific aspects here that I probably don't want to bore you with. But if somebody looks at this and has questions specifically regarding a program that they're working on, I'm happy to answer questions later. So, okay, now I decided to start Mahzi Therapeutics to help patients develop therapies and take them into the clinic. I went to venture capital firms and started pitching to them. Now, what would convince someone to invest in rare neurodevelopmental disorders? the first thing I told them is they're underdiagnosed. We will find more patients. That is a given. There's no question about it. And luckily, they believed me. The second thing I needed to show them is that we have a few candidate therapies, and these candidates have a potential to be successful in humans. And how do we do it? We either look at cells in a tissue culture plate and test the drug on these cells, or if there is a good mouse model, and Dan, where's Dan? Dan is the expert in telling us if the drug is going to work in the mouse or not. We run some tests in the mouse to see if we're fixing what's missing in the mouse. So what is proof of concept? What do we consider as something that could potentially become a drug? And what do investors think is convincing to say, oh, maybe it's worth putting our money here because maybe there will be success and we will see a return on our investment. So there's not a one-size-fits-all. It's on a case-by-case basis. But ideally, we want to see data from cell lines, and we want to see some mouse data if there is a good mouse model available. The cells, and John just gave his cells for experiments on his daughter's mutation, we need to have cell lines that represent the patients. So either cells directly from the patients or cells that have been edited to contain the same mutation that the patient has. Because we need to see something that's representative. We can't fix anything. We need to really focus on a specific question that we have and try to fix that. So it would be the patient's cells, the patient's mutation, and cells from the parents or the brothers and sisters that have the similar background, a similar genetic background, but do not have the mutation. We need a control. And then now there's a wonderful system that maybe some of you have heard of, the scientists for sure. There are brain organoids, which are really helpful. So scientists take the cells and develop them into little mini brains that contain all the cells that are in the brain. And we can test those little structures as a model for a human brain. So it's not, these are made from human cells, and not mouse. So you have a human model, and then you can test it also in a mouse model if you have one that's available. So this is what we had for our first disorder. Again, this is an example, a depiction of what we showed investors, and they chose to invest in our company. We had what we call proof of concept in an animal model. This disorder is a very rare epileptic encephalopathy. The kid sees hundreds of times a day. Extremely fragile kids. They die very early on because of complications of their seizures. And we were very lucky that the mouse model, and you see the mouse in the photo, the mouse model is very similar in its characteristics to the humans. The mice seize, they do not develop, they die by the age of 3 weeks. Mice can live for years, just for reference. They die by the age of 3 weeks. They have many systemic abnormalities. They have hyperlipidemia, so they have high cholesterol. So they have a lot of things that we can measure to see if they change when we give a therapy. So if you look at the mice, the mouse in the center is the mouse model of the disease. The mouse on the left-hand side is a normal mouse. For reference, the same age, but developed obviously normally versus the mouse that has the mutated gene in it. And on the right-hand side is the mouse that received treatment to show that the mouse actually continued to grow and develop like the wild type, like the normal mouse, and did not die like the diseased mouse. So what we were able to show, and in this case it's a gene therapy, so we put the gene into the virus that I mentioned, the AAV virus, and we put it into the brains of the mice. We were able to show that once we put the gene therapy and it made the normal protein back, it actually normalized the mice that were injected. You can see they didn't die in the graphs. You can see that they stopped seizing. You can see that they lived longer. We basically normalized them. So this was good proof to us to say, hey, this is a therapy that we shouldn't leave on a shelf somewhere. This actually can get into kids and maybe change the trajectory of their lives. What happens after we have proof of concept? So the proof of concept is only the beginning. The path is still very long until we get to a program to a product that gets into human beings, into clinical trials. So this is just the whole process of drug discovery. If you look at things, I talked about research development, I talked about the preclinical studies. Now let's move into how do we get into the clinical studies? So it's a path that now we get to involve the regulators. The regulators, they, of course, care if the drug works, but their main concern is the safety of the drug. So we have to do a whole set of experiments that actually proves that the drug not only works, but is also safe. And unfortunately, safe in mice is not enough. Sometimes we have to show that it's safe in higher organisms as well. And these are studies that are very well regulated. They're like a recipe book. Do X, Y, and Z. Come to us when you finished X, Y, and Z. And if you don't have any deaths or any serious adverse events, we'll let you dose people now. So this is a little bit of a different depiction of the same path. On the left-hand side, you see everything that we did up to the safety studies. And then the last part, which is in the red square, is the IND-enabling studies. IND is an investigational new drug. That's what you file to the FDA to get approval to start dosing human beings. And it's a whole set of experiments that takes more than a year to conduct and to show that the drug is safe. It distributes it, it gets to the cells that it needs to get to, and that we know how much of it we want to give. That we're not going to underdose, because especially in gene therapy, it's an invasive procedure. If we underdose and we don't give enough, that's it. The patient cannot come back again. That's their only chance. And if we overdose, obviously, we can cause some adverse events. So we need to be very clear about what the dose range is. So do we take 1 pill of aspirin a day or 5 pills of aspirin a day? That's easy. But when it comes to genetic-based therapies, you have to be a lot more precise on the dose that you're planning to give. So the goals of the IND package, as I said, proof of concept, characterize the drug, understand better what it does, where it goes, how it gets eliminated. When it comes to viruses, obviously, you know there's scares that the viruses will be eliminated through feces and maybe transfect the whole town. No, it's not going to happen, but we have to show that it's not going to happen. And then, as I said before, it's just the first part of the post-proof of concept journey. Now we need to take it into clinical studies. Clinical development. So in our case, since most of these disorders are very rare, they've never been in a clinical trial. I go back to obesity because that's like the flavor of the day for investors these days. Obesity, yeah, it's been in 1,000 clinical trials. You know what the endpoint is. I started in weight X and I ended in weight Y. For neurodevelopmental disorders, they've never been in clinical studies before. It's very hard to assess progress if you have a child that doesn't say one word. How do you measure gaining 1 word or 2 words, and which words, and how these words can make a difference in the family's life? You have to actually engineer a whole set of experiments that will teach us how to understand progress of the patients. And we have to validate them, and we have to go to the FDA and say, we validated this. We know it's valid, and we know that every patient that will get our drug will now show improvement in this, and we can measure the improvement. And it's not easy, especially with neurodevelopmental disorders. And especially with disease-modifying therapies that we hope will have a lasting effect on the patients. How do we measure this lasting effect? So as I said, the challenge is validating these new endpoints. Another challenge is that since there's so few patients, the studies are very small. Can you show statistical significance? Or are you hoping that the change will be so robust that you just show a video to someone, the kid with the SMA, right? There's a very famous video. I think no one remembers what the results of the clinical trial were exactly, but there's a famous video of a little child with a backpack running in the corridor. And that child would have not been able to do that if he had not been treated. So how can we find these endpoints that are actually very visual, that anyone that looks at the child that knew the child before and got the therapy after the therapy would say, wow, the therapy really made an impact. So we're not looking for statistical significant endpoints. We're looking for really significant endpoints for the family and for the patients. And how do we take everyone's experience? We asked John in Camden, what is the most important thing that you'd like to see changed after a therapy?
Some toileting.
Toileting, okay. Potty training, right? How do you measure potty training in a clinical trial? How do you do that? It hasn't been done before. We have to figure out how to do that. And we have to go to the FDA and prove to the FDA that toileting is actually worth an endpoint that is worth the whole investigation, and then go to the insurance and say, Camden now can use the bathroom on her own. Will you reimburse that drug? That's a challenge. It's not something that's so self-evident. For John, it could change the life of the family, but when you go to insurer that never experienced these disorders before, how do you explain to them what is valuable? So for each disorder that we take on now, we have to start from zero and build a story and hope that it will stick. And hopefully the more successes that we see in the neurodevelopment field, we'll be able to convince the payers, the regulators, even the clinicians. Some of them think, oh, toileting, that's not a thing. I want to see the child running a marathon. Yeah, thank you, that's never going to happen. So we need to work on making sure that everyone is aligned when we develop endpoints. And another thing that I won't go into is just because it's really important for us, is can we find a way to measure that the drug is working quickly? To know it's working, it's worthwhile to continue to treating kids, it's worthwhile continuing to dose within weeks. And we're looking for different elements in the blood or in the cerebrospinal fluid to see if we can find something that will indicate that the drug's working quickly. And then a lot of regulatory, you heard me say FDA way too many times, but that's the secret. Working with hand in hand with the regulators and making sure that you really have a path to approve a drug. One thing that we spend a lot of time, and I said that things are done in parallel. So while Paul and Dan and the team here are working on a therapy for Camden, in parallel, we need to understand how do these kids normally present? Some of them are able to make gains over time, but they're very slow and they're not like a normal child. Others don't make gains at all, and some even deteriorate. So how do we know what a drug is doing if we don't know what happens to the patients naturally? So we kick off a natural history study that starts with interviewing caregivers and asking them, just like I asked John, what would you like to see changed? We then go and we interview a lot of families to understand that. We interview clinicians, and at the end, we actually take all the feedback that we have and start measuring all these stories that we were told that I want to see speech, I want to see toileting. We put it on a time scale. What can a 1-year-old do? What can a 3-year-old do? What can a 5-year-old do? And then when we treat a 5-year-old, we actually know what improvement would look like in that 5-year-old. So this is something that we do in parallel. If we wait till there's a therapy and then we start doing this, we lose a lot of time. So this has to be done sooner better than later. And the last part that I wanted to talk about, and this is really the reason that I'm here, because of John and his story and his efforts to develop a therapy for Camden, is how we work with patient groups. And as you saw before, there are hundreds of new genes, which means that there are hundreds of new patient groups. And each patient group is now trying to develop a therapy, not waiting for biotech or pharma. They're doing it. So how do we make sure that this becomes an efficient process? So if you look on the left-hand side, there's 3 groups that if you know a little bit about what's going on in the pharmaceutical industry, you would recognize these names. One is Cure Duchenne. It's a group that has funded a lot of work for Duchenne muscular dystrophy, has invested in companies, saw returns on their investments, and now they're a very, very rich group that can continue to develop more therapies for their disorder. The same for the Cystic Fibrosis Foundation. Cystic fibrosis didn't have a cure until probably 10 years ago. Cystic Fibrosis Foundation funded research, enabled drugs to be developed, and are now seeing not only a drug for their patients, but also more funding to invest in additional and new drugs. And on the left-hand side, which is relevant for neurodevelopment, FAST is the Angelman Foundation. Angelman is a neurodevelopmental disorder. The group started a company, sold it to another company, and now is using the proceeds to develop more therapies. So how do we replicate these very successful groups? How do we help other groups? On the right-hand side, you'll see every gene almost has a group now. How do we help these very motivated groups How do we guide them to develop the right work plan to get a drug into proof of concept in an animal or cells and take it into the clinic? So talking about parallel tracking, these are the pillars that I usually talk about. On the left-right side is the community. What can the community bring to the table? On the left-hand side is the disease understanding. What do the scientists bring to the table? What are the tools that we need in order to develop a therapy? And what are the therapies themselves that we are looking for? The community brings they are the most knowledgeable people about on the disorder. They're the ones that are going to tell us, this is what my child does on a daily basis. This is what the child cannot do. They're going to participate in natural history studies and provide all the information that we need in order to design a clinical trial. On the left-hand side, we will talk to clinicians, we will talk to the scientists and the parents and design endpoints for clinical trials so that we know what we're measuring. We'll look for biomarkers and we'll define the disorder and try to understand it better. The tools I talked about would be patient cells, organoids, a bank of tissues if it's available, and develop the relevant animal model for proof of concept. And on the left-hand side, what I talked about before is what is proof of concept, how do we choose the modalities, and what do we do in order to develop the therapies and make sure that they get into the patients. Parallel tracking, off the bat, John started a foundation. In theory, what he should have done, day 0, come to this building, give his cells, make sure that they're IPS cells from him and from his family and from Camden. Understand the disease biology, what you're doing already. Starting to develop a therapy and show that the therapy is working, and then transition, as I said before, to IND-enabling studies. In parallel, make sure that more patients are diagnosed. Start a Facebook page. I know you did all of this. Start a Facebook page. Reach out to all diagnostics labs. Ask, how many patients have you diagnosed? How will you help me diagnose more patients? That should start in the very beginning, because by the end of the process, maybe you'll have enough patients for a clinical study. And then natural history, as I said, also start it before you have anything that is related to a drug, because you'll need this information regardless when you get to clinical trials. And then on top, the top line is the clinical endpoints. What are we going to measure? We have a protocol. We're going to the FDA to ask if they approve our protocol. This is what we want to measure. This is how we're going to do it. Are you agreeing to what we're proposing? If they say yes, then we go look for sites. Who is going to actually execute the study? Who knows how to do a gene therapy study? Who knows how to inject gene therapy into the brain? Who knows how to inject an ASO into the spine? Who has the expertise? Who wants to learn? We'll select sites that know how to do it, that can recruit patients, and go into clinical trials. So going back to the Mahzi story, and I'm almost at the end, so I want to leave some time for questions. You'll see the big green arrow, which starts with the advocacy groups funding academics. That's how it all starts. Your child gets a diagnosis, you Google the name of the gene, you Google the name of the disorder, you find the scientist that published the most papers on your gene, you reach out to that scientist and ask, are you willing to work with me to develop a therapy? Patient groups raise money, bake sales, golf tournaments, galas, whatever. They use this money to fund the academic research. Proof of concept is achieved. Mahzi comes in, or companies like Mahzi. We take over the program. We're the professionals. We know how to run the safety studies that I mentioned. We know how to design clinical trials, and we take things into the clinic. So everything needs to be aligned. The advocacy groups are, as I said, are the experts. They provide all the information. The academics are the experts in the science, but there needs to be alignment. You can't think that you're going to write a grant, send it to the NIH, and get an answer in 2 years if you got the grant or not. It's not going to work. The clock's ticking. You need to find other mechanisms, other ways to fund research. And if you're taking money from an advocacy group, you're accountable. You have to adopt their sense of urgency and make sure that you're meeting the milestones and developing the drug that you promised that you will. And only if there is alignment between the advocacy groups and the academics, and eventually a company like ours, we will be able to get therapies to the markets. So in summary, it takes a village to develop a therapy. It won't work without collaboration and synergies. And the keys to success are really the solid biology that you start with, smooth handovers between the different parties that are participating in this process, parallel tracking, not trying to do one after the other, but do everything as much as you can in parallel and collaborate. Thank you. Happy to take any questions.
So we have a number of great questions online. The first one is, are there any examples where organoids contributed to treatment development?
So yes, definitely. We have an example. We're working on a disorder called Pitt-Hopkins. And the work started in a lab at UCSD that has organoids from Pitt-Hopkins patients and their families. And they first tested their gene therapy in the organoids and showed that there was a successful reversal of whatever was observed in the organoids. And from there, we took it into a mouse model, but the organoid work was very convincing and made us really want to take over this program and develop it.
What are the specific criteria for advancing a drug candidate from phase 2 clinical trials to phase 3, and how does the sample size change between these stages?
Well, in general, phase 1-2 is the time in the clinical development where you determine the dose, where you validate your endpoints, and you want to be sure that you're testing the right things when you go into the larger phase 3. However, in rare disease, the path is a little different. You can actually likely run a phase 1, 2, 3. You learn as you go, and you can run a seamless study, but you still need to design it in a robust enough way to make sure that at the end of the last phase of however you defined it, you have proof that the drug is working. So in general, I think for a small molecule or any other drug that treats many patients, the phase 1-2 is usually small, and the phase 3 is huge. In rare disease, there are exceptions.
Do patient groups usually help on getting data for proof of concept in human cells and mice? And so what are they usually looking for in these projects?
So yeah, there are many patient groups that do it, and they're looking for something that will convince me to take over the program, basically, that the drug is working, that there is some way to show that the drug is working.
It's not a barrier. It would be preclinical work.
No, not at all. Definitely not for us.
Is there a patient sample size that triggers industry interest, a theoretical threshold?
It depends on the modality. For gene therapy, the thresholds are a little lower because gene therapies, I don't know if you've heard, but they cost a lot of money. They're $3-4 million per patient, so you can recover your expenses a little faster with a smaller number of patients. But for small molecules or even antisense oligonucleotides, probably thousands is the bar, thousands of patients.
Okay. We have a couple of questions about using.
Maybe if there's anyone in the audience that wants to ask questions before we do the online?
We do it all.
Okay. You do it all online. So you're reading people that's sitting here?
They've been sending.
But I want to see who's asking what. Like, okay, stand up. It's not very personal this way.
How can these strategies be adapted to multi-gene large deletions?
I think gene editing will likely be the tool for the large deletions, but specifically for the brain, we still don't have tools to edit in the brain, so it's still a way to go.
Okay. And then kind of similarly, are there ways to use gene therapy for gain of function mutation?
So gene therapy, so to restore the gene for gain of function, it depends. In some cases, you need to knock down the gene, the mutated gene, because it's doing something that's interfering, but you need to replace and restore what's missing because 50% is not enough. So in this case, it will be a combination of knocking down and adding. Just for gain of function, probably gene therapy is not the right modality.
That's about the online question.
Okay, now if anyone is brave enough to ask a question in person, let's do.
So one question I have is, say, you're affected by a disease or your family member or friend is affected by a disease that is extremely rare. So what's the path forward there? Yeah, sure, finding more patients, but sometimes that's not an option. So like, what options are out there? What would you suggest somebody do if they've tried all these things and they can't find other people? It might just be a challenge, but do you have any thoughts?
So the question is whether it's a unique mutation or a unique gene, because
Just say theoretically, it's a unique problem, whatever it is.
so there is an effort today that is called the N of One Collaborative, and it's actually aimed at developing treatments for specific patients that have a very unique mutation or a gene that is mutated in a very, very low frequency. And there are a few mechanisms. There are several universities that are working specifically on N of one mutations. One of them is St. Jude's. There's another one in, I think, in the Netherlands. There are a few centers that have taken on to develop single patient therapies. I think the biggest problem is the moment there is a therapy, you find out there are actually a lot more patients than just the one that you thought there is. And then how do these patients get access to what's already been developed? I think that's the biggest challenge at the moment. There's also a non-for-profit called NLOREM, and they make it their business to develop therapies for what they call nano-rare patients. That's another resource. But again, you find out that you're not the only one, which is great, but you need more, you need to get resources to treat more patients than just one.
Well, I really want to thank you for your work and for sharing your work. And as a token of our appreciation, this represents trying to improve health for individuals in Missouri using precision approaches as you're demonstrating here. And
Thank you very
also
much.
want to thank John Ruth and Paul Carney for growing this effort. This is a very exciting project, and it really aligns well with what we're really trying to do with NextGen Precision Health. So thank you so much.
Thank you for having me. Really a pleasure. Thank you.
So I do want to just make one more announcement for the next Discovery Series and also the NextGen Fellows. So do remember to apply by October 15th. And please join us for Dr. Mark Howler when he presents on October 2nd. Thank you.